Welcome to lesson 4.4, skill 4 in action. This is the last of the 4 skills that the AAMC has identified as common to all of the sections of the MCAT, and chapter 4 was all about skill 4. In this lesson, we're going to go back over a lot of the principles that you've learned and pull them together in a way that earns points on test day. You're going to see that skill 4 is tightly linked to skill 3. I'm Tyler Farah. Let's begin. See what the test makers have to say about skill 4 questions. They start by making a direct comparison to skill 3 questions because both question types ask how you, quote, do science. So I'd like you to take a second now, read over this paragraph, and make a note of how skill 4 differs from skill 3. Skill 4 is all about data analysis. This is where things like mean and median and mode, standard deviation, analysis of graphs, and those kinds of things are going to be tested. Also, it's super important to notice that drawing conclusions based on graphs, figures, tables, and so on, that's all part of this skill. So you can bet when you see a passage with an experiment, at least one of the questions is going to ask you to draw a conclusion about that experiment based on the data that's given. So to see this skill in action and to review all of chapter 4, we're going to apply measures of central tendency and dispersion to data sets. We're going to look at visual interpretations of data, and we're going to see a couple of questions about error as well. And we'll do all of this by looking at a passage and question set that's all about data analysis for a pretty complicated experiment. The underlying science for this lesson is going to be molecular genetics and the cell cycle. So without further ado, let's look at our first learning goal, applying measures of central tendency and dispersion. As you read this passage, you'll probably actually recognize the topic from lesson 3.4, the review of chapter 3. In fact, you could even consider this passage to be the second half of the passage from that lesson. But if you haven't read lesson 3.4, or if it's been a while, don't worry. This passage works as a freestanding passage in its own right. So take a moment to read and outline this passage now, and don't forget that in your book, this passage continues on for one more paragraph and a table. Go ahead and read and outline this now. This passage presents the results from a CHIP assay. And this assay examines a molecule called cohesin, which the experimenters claim holds sister chromatids together. But this passage isn't really about stuff that you don't know, like cohesins and CHIP assays. This is really about stuff that you do know. So think for a second. When do sister chromatids need to be held together? Well, sister chromatids are produced by replication in S phase, and then they separate at the metaphase-anaphase transition in mitosis. So the data presentation here is pretty busy. Our scientist has looked at a few different binding sites, and she's run everything in triplicate, so there's just a ton of data here. But look at the axes. She seems to be making these measurements with time in the cell cycle. And it does seem that at least for binding site 1, there's a pretty clear spike from S phase up to metaphase. So the real takeaway here is that the MCAT doesn't expect you to know details about cohesins and chromatin immunoprecipitations. The test makers expect you to know about the cell cycle, sister chromatids, and their separation in mitosis. And then with the table, the test makers are really just presenting similar information in a different format. These data aren't the exact same as the ones in the figure, but they do show a really similar trend. Again, thinking through some basics about the cell cycle first helps interpret these data. So now let's earn some points. For this first question, I'm going to give you an unfiltered go at it. That is, I'll just let you answer the question right now on your own. And here's table 1 from the passage for your reference. Go ahead and try this question right now. Now, what I love about this question is that it's not so hard, but it actually teaches you something really cool about measures of central tendency, aka mean and median. After all, you'll never see this question again, so you might as well learn something from this question for the future. The question references table 1. So I hope you started with table 1, and it's looking at prophase specifically. Now, within the data for prophase, the median is really easy to find because it's just the number in the middle right here. That's going to be our median. And it's really important with this question to recognize it doesn't actually ask you to find the mean. It asks you to compare the mean to the median. So you certainly could do that by calculating the mean, but you don't have to. Remember, the mean is the mathematical average of these three numbers. So where the median just kind of threw out the biggest and smallest numbers and really just looked at the middle, the mean is going to incorporate all three numbers into its calculation. So it's because the mean uses all of the data, including the extreme data points, that the mean is more sensitive to extreme data points. All this means is that the mean is going to be dragged up by that very large data point. This 63 here is going to cause the mean to end up to be larger than the median. Now, if you did do that calculation, you'd find that the mean was 54, which is greater than the median at 50. But either way, the takeaway from this is that the mean is a measure of central tendency that's more sensitive to extreme data points. The median, by contrast, is a different measure of central tendency that sort of ignores extreme data. Our second question looks again at table 1, and this time it's asking us about standard deviation. Now, I'll be honest, I dread calculating standard deviation. The formula is so time-consuming, and it means spending like 2 precious minutes on test day doing arithmetic. So fortunately, with this question, use the data and see if you can find the answer without doing any calculations. So to find the trial with the highest standard deviation, but without calculating, think first about what standard deviation is. It's a measure of dispersion in the data set. That is, how spread out the data is. There are other measures of dispersion, but standard deviation is definitely the most widely used. So really this question is asking, which phase has the most spread out data? And here, of the answer choices, S phase has the most spread out data and also the highest standard deviation. What you've just seen is that on the MCAT, data analysis need not necessarily mean dusty, challenging, rote calculations. In fact, a lot of the time, understanding the principles behind the calculations is enough to get the points without doing any calculations. In the next portion of this lesson, we're going to take a look at another related data analysis skill, understanding visual interpretations of data. In this learning goal, we're going to study the best ways to organize and interpret data, whether it be in a chart or a table, any kind of visual representation of data. Question 3 is a really fun one. Here, we're taking data from the table, and we'll be turning it into a bar graph format. Notice that none of the answer choices, though, actually gives numbers on the y-axis. This is all about a qualitative comparison of these data, so it's all about trends. Take a second and answer this question, and be sure to consider the prompt. What trends are even present in table 1? This is the exact kind of question that you'll ask yourself during the execute step for a question like this on test day, so use that guidance to help you out on this question. I've provided the data from the passage in the table for your reference. Go ahead and answer the prompt and question 3 right now. Let's spend some time analyzing these data. First, cohesin binding seems to spike up during S phase, but that's actually reflected in all of the answer choices, so that observation is a good start, but it doesn't get us any closer to the answer. The next observation, though, is that from S phase to G2, the numbers seem to be actually pretty close together, and we can use that to rule out letter A, because letter A has a pretty sizable jump from S phase up to G2. Now, also, it looks like even though the numbers are similar from S phase to G2, point for point, each number is higher in G2 than in S phase. So the peaks in G2 really should be the highest peaks overall. That allows us to rule out letter B, because letter B actually shows a drop from S phase down to G2. And then the final observation is that going from G2 to prophase, there's a pretty substantial drop. All those numbers get cut in half or more. And that allows us to knock out answer choice C, because in answer choice C, there really isn't an appreciable drop from G2 to prophase. That leaves us with answer choice D, which is the right answer. This one shows the jump from G phase to S1. It shows that G2 is just a little higher than S phase. And then it shows a drop from G2 down to P phase as shown in the table itself. So again, this skill of connecting different representations of data and drawing conclusions from data, that's what skill 4 is all about. And question 4 goes in this direction as well. Here we are literally asked to make a conclusion based on the data. The question asks about binding region 1. So the first thing you'll need to do is figure out which set of data to even use to answer this question. But go ahead and answer the prompt on screen and the question right now. Binding region 1 was found in the figure. And one key to this problem is to sort of mentally filter out all of the extraneous data that's shown in this figure. All that really matters is the set of darkly shaded bars, since those correspond to binding region 1. And what you see here is a trend that's very similar to the data in the table. First off, we have a spike in cohesin levels from G1 to S phase. Cohesin levels seem to remain elevated throughout S phase, G2, prophase, and metaphase. And then at the metaphase anaphase transition, there is a precipitous drop in cohesin binding levels. Now, since these data represent cohesin binding, and since the passage says cohesin holds chromatids together, this trend supports the conclusion that sister chromatids detach after metaphase, since there's no more cohesin to hold them together. The wrong answer choices here, though, probably teach just as much as the right answer, though. For choice A, it's really easy to get sucked into the new information that's given about cohesin and how it relates to the cell cycle. But instead, focus on the end of this answer. A basic fact about sister chromatids is that they are created in replication during S phase. So in G1, there's just one set of genes. Sister chromatids just don't exist. That makes A scientifically impossible. And recognizing that this question, like most questions, is not about advanced science that's outside the scope of the MCAT is going to help you rule out answer choice A. For B, the data in the table is actually of a totally different kind than the data in the figure. That is, the figure looks at cohesin binding at specific binding sites, where the table in the passage looks at total binding overall. So because the data is actually of different types, specific sites versus overall binding, it's actually not possible for it to be in conflict. Choice B can therefore be avoided by paying close attention to the data in the figure and in the table. And for choice C, the passage tells us that there's actually 3 binding regions on a given yeast chromosome. So our scientist doesn't take care to control for which of the sister chromatids is being bound. Again, deciphering the legend of the table helps us avoid choice C. So this question overall really was all about thinking about how the data in this passage relates to science that you already know or conclusions that can be drawn from this experiment. In our third learning goal, we'd like to take a look at something that is routinely tested on the MCAT, comparing and contrasting different types of error. Question 5 may not come right out and say it, but this question really hinges on determining the type of error. After all, it asks about reducing the error, and to reduce the error, you've got to first know what kind of error you have. The other thing to be careful of here is the not in the question stem. I'll leave you with that warning, but go ahead and try out question 5 right now. The type of error in this experiment is random error. And the reason why is that a given yeast cell can move faster through the cell cycle or slower. So the error isn't consistent, it's random. So what then is the cure for random error? Well, increasing the sample size will help eliminate random error, since each cell that moves too fast will be matched by a cell that moves too slow. So the randomness averages out with a large enough sample. The other tricky part of this question is the not in the question stem. What that tells you is that if something represents an increase in the sample size, then it will reduce random error. And that makes the answer wrong. After thinking it through like that, in your execute step, you might actually rephrase this question as, which of the following is not a way to increase the sample size? Even then, though, the MCAT has one last trick here, which is that actually every one of these answer choices increases something. But choice C increases the sensitivity of the measurement device, not the sample size. So the answer here is choice C. And our last question of this set is, again, all about identifying error. Identify the error type described in this question and answer the question right now. The key word here is consistent. This word is a signal for systematic error. Systematic error is introduced by the experimental procedure or by an experimental device, and it is identifiable by the fact that it is usually consistently above or below the real value. For example, a scale that isn't zeroed properly will always weigh patients wrong, but it will always weigh them wrong in the same way or by the same amount. In fact, sources of systematic error can actually be pretty hard to spot because the error will be the same from trial to trial, and so it may not appear that there actually is any error present at all. In any case, the answer to this question is letter B, systematic error. The only somewhat tricky part of this question is that the test makers are hoping you'll see the bit about under-reporting for one site and over-reporting for another site, and they're hoping you'll think random error. This is because random error does have that pattern of sometimes being too high and sometimes being too low. The key difference is that random error is randomly high or low, so it will change from trial to trial. This question describes a faulty device that is consistently high or consistently low. But this does go to show systematic error can be a little more complicated than initially meets the eye. For example, you may have used a pH meter that needs to be calibrated before use. Upon calibration, that pH meter should have no error. But as you use the pH probe in different solutions, it becomes less and less accurate, introducing more and more error, unless you periodically recalibrate the probe. So that would be an example of systematic error actually changing over time. You might think of it like this, though. Consistent error is systematic error, although not all systematic error is consistent. So in short, the fact that error here is consistent and is due to a faulty experimental device, that tells you this is systematic error. And as review, we've listed out all of the learning goals from Chapter 4 as a whole. Keep in mind, Skill 4 is all about data. You see analyzing and interpreting representations of data, choosing the most appropriate representation of data, using data to identify and explain relationships between variables. You get the point. Interpreting data, applying measures of central tendency like mean and median, or of dispersion like standard deviation. These are skills that are directly tested on the MCAT. So it's worth studying experimental design and data analysis right alongside studying the science content of this test. So that concludes lesson 4.4, which was all about Skill 4 data analysis. And that also means concluding the lessons on the 4 science skills in general. Remember, these 4 skills, content, critical thinking, experimental design, and data analysis, are the 4 keys to your success on the science sections. So be sure to study all of them equally, find your weak areas, and turn them into strengths.